Today we're going to be talking about the identification and sequencing of the self-splicing introns from wild ciliate species in the Pacific Northwest. My name is Connor Shiomoto,
I'm Mason Nakamura.
and we'd like to thank our advisor, Dr. John Schnorr, here at Pacific University for all of his help in our research.
Tetrahymena thermophila. They are single-celled eukaryotes, and a paper written by Riel and colleagues describe them as model organisms. We call them model organisms because they're easy to grow and they're easy to find in the wild and they're easy to isolate in a lab setting. And since we're in the Pacific Northwest, it makes them very easy to find in freshwater.
So some things that we've learned from studying Tetrahymena thermophila. There was actually a Nobel Prize in 1989 awarded to Altman and Czech for their discovery of catalytic properties of RNA. Czech was actually studying Tetrahymena thermophila when he discovered that an unprocessed RNA molecule, when placed into a test tube, had the ability to splice itself. So in other words, RNA molecules could cut themselves into pieces and then join the genetically important RNA fragments together again. Um, Czech was the first to really demonstrate that RNA was capable of conducting its own chemistry. And this really changed our understanding of the molecular basis of life. Because prior to that, everyone just thought that enzymatic properties were exclusively in proteins.
On the left, we have a picture of the central dogma, which is eukaryotic DNA replication, where the DNA in the nucleus is transcribed into RNA, which is then converted into mRNA, in which non-coding regions of the DNA are spliced out by a spliceosome. And if you look at RNA, the green bands are the introns, and the introns are non-coding regions. And then the red bands are exons, which are coding regions. And when the coding regions recombine and leave the nucleus, it's called mRNA, and then mRNA is translated into amino acids and then are folded into proteins. And on the right is going to be talking more about RNA, where the exons and introns are shown. And in the middle, a spliceosome attaches to the intron and splices it out. Then the exons recombine into mature mRNA. And then on the right, it shows an example where there's no spliceosome or protein that attaches to the intron. Instead, the intron takes itself out, which is called self-splicing. And then when the two blue bands, the exons recombine, they recombine in different ways when the intron leaves, and this diversifies protein output.
So what Czech really saw in 1989 was basically these self-splicing introns, or otherwise known as SSI, which are basically non-protein coding regions of RNA nucleotides that are able to conduct their own chemistry in which they remove themselves from the genome without the help of any other proteins. So you'll see it's splitting these two exons, and then the intron will essentially remove itself and then leave behind only the spliced exons. And this was the first time a real stretch of like RNA had been like identified to have its own catalytic capability. And when Czech was studying them in Tetrahymena thermophila, he actually discovered a group 1 intron, which is found throughout all kinds of Tetrahymena species. There's also other groups like group 2 and group 3, which are more common in archaea and plants. But today we're going to be specifically looking at the group 1 intron in Tetrahymena.
Previous research on tetrahymena and SSI. So Dorder and his colleagues in 2018 did a massive study on ciliates, specifically tetrahymena, and tested them for SSI presence. On the left is a map showing all the dots are where all these mapped and found their ciliates. On the right is a phylogeny tree. And you can see on the far right, there's a split in Borealis and Australis, and those are clades. And clades mean that they belong to different groups, but they all belong to the same common ancestor. And then the circles, the clear means that they're SSI negative, and the positive is the filled in with black. And then in some examples, you can see T. grucci in the middle. It's mixed. So that means when you find it in the wild, it could be either negative or it could be positive. And then what they found was they had 13 positive SSIs, five mixed haplotypes, which is plus and negative, and 27 negative overall. And then it just shows that there's variation in SSI presence, and some species are going to be positive and negative mixed.
There's essentially two ideas that could potentially explain the variation of SSI and tetrahymena. The first being that an ancestor of Borealis and Australis acquired the SSI in the large ribosomal subunit, and then when it speciated and created the Borealis and Australis clades, all of those subspecies would have had the SSI with them unless it was lost. The more commonly accepted idea under prior research done by Paul Dorter is that the current SSI acquisition in tetrahymena species reflects two independent acquisitions through horizontal gene transfer in the genome when they had already split. So Borealis and Australis had already been speciated, and then they acquired the self-splicing intron. So that would explain why there's so many SSI negatives and some SSI mix. Basically just means that these acquisitions have been evolving independently of each other in the SSI, and those mutations continue to be passed down, and we're able to map that through the SSI gene and show that there really is a relationship that supports that this independent acquisition happened twice.
Questions that we want to go over. So we wanted to test what tetrahymena species we can find in the Pacific Northwest. If the pattern is going to be the same with the East, if the SSIs are the same or if they're different, and if we could identify any new species with SSI in the Pacific Northwest. And then for methods, for sampling, on the left, we have a photo from Aldous and Kiera, and they're in Scappoose Bay, I think. And for sampling, we use a golf ball retriever and tie a Ziploc bag to it, and we scoop some water. And we then add, we want to grow that water, and hopefully tetrahymena or ciliates are inside. We use penicillin and streptomycin, which slows bacterial growth, normicin, which targets and eliminates slower growing bacteria. And then we have SSP media, which is rich media that favors tetrahymena, which will help it grow and give it nutrients. And then amphoteric B is a fungal, it eliminates fungal growth overall. And then on the right is clonal isolation. So we use these wells where we add all those things on the left right here onto. And we add little droplets from our bags, hopefully that we could isolate one cell of tetrahymena, and then it'll grow into a whole population of a specific species so we can isolate it and study it. And then you can see some of the wells are kind of cloudy. Those are just fungus growth, so that's a failure of a well, and that's not what you want to see. And after we isolate something from the wells, like tetrahymena cells, we add that to Chelex, which is used for amplification and purification. What Chelex does, it makes sure that the DNA doesn't get broken down. So on the photo on the right, you can see DNA is at the top of the water, and then Chelex is going to be in the bottom. And then we could use that DNA and transfer it out. So then we could amplify it to see it in the COX-1 sequence and in the SSI sequence.
So after the environmental DNA was basically extracted from our long-term cultures, when we were designing our primers for the SSI, we actually designed them based on the alignment of 8 close homologs of tetrahymena and other ciliate species and uncultured eukaryotes. And these sequences all span the SSI and the large ribosomal subunit where that self-splicing intron inserts itself. And basically the way that we designed these primers was so that the PCR amplification would allow us to detect the presence of the SSI based on the size of the bands. So our forward primer is right here and our reverse primer is right here. And then this would be the large ribosomal subunit gene on the bottom. So even if there's no self-splicing intron present, we would still have 250 base pairs regardless when we run them on a gel. So T-Shang, which is an identified SSI negative genome, it'll still show up on a gel compared to a negative where you don't see anything. So it still shows that we had PCR amplification, but it also doesn't show that we had the 400 or the 650 base pairs that you would find for an SSI positive genome. So if the SSI, which is 400 base pairs roughly, was present, you would see a 650 base pair band right here, like Tetrahymena thermophila, which is a known SSI positive because that's the first one that Czech identified the self-splicing intron in. And that should be around 600, roughly 650 base pairs.
What we tested and what we found. So we tested 20 isolates for SSI, and it ended up being only 13 species. But we did find 5 positive SSI, and that 3 isolates came from T. pigmentosa, which was found in Fern Hill. And they were identical to the ones that we found on the East Coast. And we found 1 SSI positive of T. aquafluente. And then next slide shows the gels where we find a positive. So on the left is the NEB ladder, and this shows the base pairs. And then we use the base pairs to see if it's positive or negative. In the red, you can see where it says 600. That's a positive hit for T. pigmentosa because it's around the 600 base pair length, where the negatives on the left are going to be around like the 200 base pair length. And on the right, we found this is our T. scapus one, which is SSI positive.
We also identified a negative control to make sure that we didn't have any sort of DNA contaminants. And we also have small primer dimers showing up in some of our gels. But the main focus is those SSI positive versus SSI negative. So after we would identify something that was SSI positive, we would actually sequence it and then create an alignment of the SSI gene based on the identification of T. pigmentosa, which was identified by its COX-1 gene. So basically, what we tested their DNA for all our classmates basically collected a sample and created 3 clonal colonies that were characterized by T. pigmentosa by their COX-1 gene. So when we then tested their DNA for the SSI, all 3 of those samples were found to be SSI positive and had indicated and had identical SSI sequences for 407 base pairs. So that those are these PNW 1 through 3, and those all show 100% homology to each other for all 407 of those base pairs. And then when we compared to Dorter's strain that was identified on the East Coast, this T. pigmentosa here, it was also 100% homologous. This means that no changes exist in the SSI on the West Coast compared to the East Coast. And this figure is an alignment that shows these 3 pigmentoses, basically, that we identified versus Dorter's. And then all matching base pairs are identified as these small little dots, and everything's color coordinated based on the nucleotide. So we basically did the exact same thing for T. aquafente, which was the other SSI positive isolate that we found here in the Pacific Northwest. And that's shown here on top. And that one's also matched up to T. aquafente that was discovered in Dorter's paper for its SSI. And this one too showed that exact same homology, 100% identical. One thing to note that this one's only 402 base pairs, whereas this one was 407. The reason that you see the difference in total number of base pairs in the SSI is because it's a non-coding region, so it doesn't actually contribute to the formation of proteins. So when you lose individual nucleotides through single nucleotide polymorphisms, whether that be like insertions or deletions, it won't actually have an effect of a frame shift because this isn't a coding region. So this is just evidence to show that like these RNA are changing and then they're still capable of cutting themselves out. We also identified one new SSI that's tentatively been named T. scapus by our lab mates, Kier and Aldis. They actually found this unique isolate here in the Pacific Northwest. And when we sampled its SSI, it actually showed very little homology to other known SSI. So looking here, these are the species and gene banks ID of other SSI positive tetrahymena species characterized by Dorter. And then this is the percent identity. So we basically created a distance matrix that was used to calculate percent identity shared between all other identified SSI and other SSI of T, like against the T. scapus. And basically the samples that we chose were based on the strains of SSI that show the closest homology to T. scapus. And Tetrahymena thermophila was also included just as like the low end and because it's what Czech initially used. But you can see here that even like everything here at the top, everything's roughly within like a tenth of a place. And so this really suggests that we do have something that's identical or it's unique. Like nothing else really shows this same SSI. So we found a new section of RNA that has its own catalytic capability, and it's still able to cut itself out. But we've found a very unique sequence that doesn't show any homology to any other self-splicing introns in the tetrahymena family. So remember that idea that the self-splicing intron is acquired twice in both Borealis and the Australis genus. Here we're looking at phylogeny of the COX-1 gene created using samples known to possess the SSI. We also included our own lab mates COX-1 sequences of the SSI positive strains. So here's T. scapus, which is under the Australis clade, T. aquafonte, which is also under the Australis clade, and T. pigmentosa, which is also under the Borealis. And then these two species marked under the purple arrows are from opposing clades and will make more sense when we look at this phylogeny of the self-splicing intron. Um, Here, looking specifically at the T. scapus, you see that it's down here and it's closely related to T. zethowini and holozi. But when you look at the COX-1 gene, you can actually see that T. holozini and zethowini are up here in the Borealis clade, where T. scapus is more in the Australis clade. So this evolution of the SSI further supports the idea that the self-splicing intron was independently acquired and suggests that it's evolving independently of the COX-1 gene and is in consistent support of Dorter's hypothesis that the self-splicing intron was independently acquired twice. And this is also reflected when you look at the COX-1 gene, when you look at alpha soalia, alpha poacea, and scolopax here in the Borealis and the Australis clades. When you look at them here under their SSI sequences, they're actually much more similarly related, which you wouldn't expect if they were under the first theory, where the SSI was evolving and everything's changing at the same rate, and it's from that initial split. So this suggests that there were two different acquisitions. Um, and then when you also look at the SSI of the ones that we characterize, like in T. pigmentosa, these show 100% homology to each other, and as do aquafonte and that was identified on the East Coast and the one that was identified here in the Pacific Northwest.
Species, and they were identical to the East Coast species found by Dorter and his colleagues. And then we found out we could say that T. pigmentosa and T. aquaflenta are cosmopolitan species because their SSIs were completely identical to the ones on the East Coast, and they're identical to the ones that we found in the Pacific Northwest. And then we also found, or Aldous and Kira found T. scapus, which is novel for COX-1 and SSI. So to further this study, we would want to do more sampling in the Pacific Northwest and hopefully find more novel species and relate it back to Dorter's paper.
Thank you guys for all listening to our presentation here today. We'd really like to acknowledge Dr. John Schnorr for all of his help in our research, and Paul Dorter, who really made this possible because of his extensive work on the East Coast of sampling in ciliates and identifying the self-splicing intron that allowed us to look here and make comparisons between the two different locations. We'd also like to thank the Pacific University Biology Department for allowing us to pursue something that we thought was really interesting. And here's our works cited. And then if you have any questions, feel free to contact us. Thank you.